Hello, everyone. We are the Genetic Engineering Society at the University of Texas at Arlington, and we are here to present our project on designing, cloning, and testing genetically engineered metabolic pathways and producing therapeutics in gut bacteria.
All right. So what is our motivation for the day? Our motivation is equal. So equal. Research has shown that equal metabolite shares a link between people with equal producing phenotype and reduced risk of certain cancers and illnesses and diseases alike, such as breast cancer, prostate cancer, and colon cancer, cardiovascular diseases, osteoporosis, and hormone-dependent illnesses as well, allowing non-equal producers to produce equal from soy products through enzymatic reactions and will allow consumers to receive the benefits of soy-derived equal. So what is equal and why is it so important? So equal is a natural isoflavone metabolite that is derived from daisy and is commonly found in soybean products including soy milk, tofu, and miso soup. Some people can naturally convert dietary daisy to equal by utilizing their gut bacteria. However, this ability is more prevalent in Asian countries than in Western. I believe there's a research shown out that Western populations, there's only around 20 to 30% that can actually naturally convert this, while Asian populations do have an advantage in a higher percentage. And as you can see on the right side of the schematic, Diazine is converted to dihydrodiazine and into equal, also producing another byproduct on the side.
The natural process of conservation of equal is a product that is derived from daisine, which is found most commonly in soybeans. With it initially coming from daisine, it is broken down through the use of its reductase, daisine reductase, or DZNR. This in turn provides dihydrodaisine that is further broken down by its reductase, DHDR, and this results into tetrahydrodaisine that is lastly reduced to equal by the use of its reductase, THDR. So here's a chart displaying what the typical process for someone that can break down daisine typically goes. So firstly, they have to digest soybeans or other legumes, and their colonic bacteria promotes the chemical breakdown of daisine through its reductases and results in equal, which also results in its health benefits of reducing certain cancers, also reducing the risk of cardiovascular diseases and osteoporosis diseases as well. The ability to produce equal is limited to a fraction of the population, but by using a engineered bacteriophage that carries these genes necessary for making the enzymes to metabolize daisine to equal, there exists the potential to re-engineer the metabolic activity of individuals. By specifically targeting the genetics in gut bacteria to promote production or increase production of DZNR, DHDR, THDR, and DDRC, which are crucial reductases in leading the conversion from daisine to equal. Through this production, it would help to reduce the risk of breast cancer, prostate cancer, and colon cancer, as well as cardiovascular diseases, osteoporosis diseases, as well as hormone-dependent illnesses. The ideal system that we would want to implement by would be through supplements. We will provide people with probiotic supplements in which the bacterial phage would administer the promotion of reductases that are necessary to break down daisine. And this would accumulate and the bacteria would grow in the gut bacteria in the gut, and large production of the reductases such as DZNR, DHDR, THDR, DDRC, and glucose dehydrogenase would be more prevalent in the person. And once they begin to eat soybeans or any products from soybeans, they'd be able to break down lysine all the way to equal and also reap the benefits of the reduction of health complications that they might face if they did not have equal.
So the creation of the plasmids. So how did we create them? So DNA plasmids were engineered through codon optimization from Lactococcus garviae strain to E. coli gene blocks. So when we actually run this experiment, we were actually able to identify that there was two different types of bacteria naturally in the human gut biome that could convert lysine to equal. However, we did choose one as it was easier to just deal with one strain to convert it, and that would be our Lactococcus garviae. So from there, cyclization of the plasmids were done through PCR using biobricks and subsequent enzymatic digests using these four restriction enzymes right here, PST1, XBAL1, EchoR1, and SPE1 of a plasmid vector, and were transformed through heat shock into DH5 alpha competent cells. So from there, we also created our bacteriophage. So the bacteriophage was cultivated within E. coli, and the phage DNA was extracted using mini-prep column purification through another set of protocols. PCR was utilized to amplify the plasmid, and DPN1 was used to digest the methylated parental DNA template. From there, the phage DNA was actually purified and cut using different restriction enzymes and purified once more to serve as the vector. You can see right here on the slide are ligation and insertion into competent cells. So PCR and digestions of the P1 and P2 plasmids, so the two Ps earlier that we mentioned, were done to amplify the desired genes inserts and then ligated when with the newly constructed FD phage vector. This modified DNA was transformed into E. coli competent cells, and then they were thus grown in LB agar plates for cultivating the desired phage. So as you can see right here, this is our, right here, this is actually the key as well for the DNA ladder when we were running the gel electrophoresis on the right side of the image. Right here, as you can see in the first wall, right here, this has our dye, our DNA ladder to be exact right here. And the DNA BP, the base pairs that we were looking for is actually 9,000 kb. So as you can see in the DNA ladder key right here, it's going to be in between, as you can see right here on the ladder itself. I don't actually, right there, between right there, just a very rough estimate, right between there, 9,000 kb. So when you can see in the second wall right here, it's actually quite close to that. However, it was hard to see with the lighting, so that was actually because of the concentration of the FD phage itself. So as you can see as well on the third wall, it's actually, you can see it's dimmer, meaning that we actually we diluted the phage, the FD tet, and we were actually run it again, and we were able to see a clear example. And then our next slide.
And here are our results from the project. So activity of plasmids was verified using thin layer chromatography or TLC. And when providing the culture cells with lysine for our P1 culture, it exhibited the hydrodysine conversion. TLC testing helps in demonstrating the effectiveness of our enzyme production, and the bacterial pellets that were producing the enzymatic pathways for L-garvales demonstrated equal presence when provided with lysine. And here is actually one of our earliest TLC plates that we have, and the P1 enzyme displays conversion of diazine to dihydrodiazine by the use of the controls of diazine, dihydrodiazine, and also equal. And the very far right P1 that is marked off here, and actually black box as well, shows that it has both presence of diazine and dihydrodiazine. And then for our most recent TLC plates, here in comparison, we have the image that you saw in the previous slide. And here's our most recent TLC plates, where we can clearly see that there is more display of equal for our newest TLC plate, and there's little to none presence of diazine or dihydrodiazine in this TLC plate, showing that our cells in the presence of diazine are able to effectively break down into equals. You're muted.
Oh, my bad. So I like to also add in a previous slide that of the two TLC, the thin layer chromatography plates, this one's actually our P1 and this one our P2. So there we received that there's different lines markings. This shows that P1 and P2 are both needed to actually properly convert diazine to equal, as you can see that there were different marks showing the conversion factors in corresponds to the levels of diazine and equal found for P1 and P2 when we ran these experiments. So my next slide, I'd like to show you the results on the gel. So this is actually our measurements, our BP base pairs for P1 and P2. For this two first sets right here is our ladder, and this one's P1. Consecutively, right here is our P2, and again, our ladder right there for easier comparison. So again, this is the same 1 kb ladder scale that we use as we did for the FD TED. And for this, we're actually looking around 2.5 kb. So as you can see right here as well, it's going to be in between here on our key. So as we look carefully on here, the screen itself, 2.5 kb, it's similar, very close to our P1 as right here, as well as our P2 right there in our results. So that was able to verify and confirm what we were looking for. Okay. So we would have our summary of the project. So overall, in conclusion, creation of a bacteriophage that can convert diazine to equal is still ongoing in our testing, as you can see from our results from our previous slide, of its efficiency, but has thus far proven successful as we are able to incorporate the necessary reductases. And these reductases clearly show the ability to convert the diazine products into equal itself.
And for our future direction, we will create a more effective phage that can more effectively produce equal and develop a supplement with this bacteriophage to be able to target specifically gut bacteria and also have it last way longer than we have currently. And we would like to quickly acknowledge both our professor, Justin Jaworski, and as well as our mentor, Priyanka, for aiding us through the experiments and just being a great help when we need help. Thank you very much for your attention. Are there any questions?
Oh, we would also like to mention as well that we had two additional teammates who were able to help us in this project. Unfortunately, they weren't able to help be here with us for our presentation today. Along as you can see on the title spade, I'm Teresa, and this is David, and our two other members that we would also like to thank will be at the beginning would be Juan Ramirez and Omar El Kashli. Thank you so much, everyone, for joining us here today and listening to our project. I hope you are intrigued, and please ask me if any questions you have on our equal products to our bacteriophages and are vectors of FD10. Thank you so much.